Hi guys, today we're going to talk about genetics of immunity and we'll get started. So kind of some of the learning objectives is we're going to talk about some of the components of the immune system. We'll look at basic blood group typing. We'll kind of look at leukocyte antigens. We'll look at some physical and innate immunity and adaptive immunity. We'll look kind of at the difference of humoral and cellular immunity, and maybe some conditions where the immune response is underactive, overactive, or misdirected. And we're going to talk a little bit about transplants. So we need to start first with foreign versus self. We know that our immune system is set up to protect organisms from foreign invaders. It provides protection, and it's based upon the ability for the body to identify the foreign molecules as what we term as non-self. Foreign things may range from bacteria to viruses, tumors, transplanted cells. Antigens are molecules that recognized by the immune system, and usually are protein, fragments, or carbohydrate. The genetics has some control over immunity. We know that genes affect immunity by conferring susceptibility or resistance to infections. There are a few types of single genes which encode for antibodies or cytokines. Genes also specify the cell surface antigens. And mutations in these genetic components can cause impairment with causing immunodeficiencies, autoimmune disorders, allergies, and even cancer. So we know that we have blood groups. We know that antigens that dot our cell surfaces determine our blood types. There are about 29 major blood types, and serology typed blood according to the red blood cell antigens. There are newer ways being developed to identify the instructions or the genes for these antigens, and they are looking at distinct specific DNA signatures when we go to type blood. And these are some of those new ways of looking at them. It's so you can see in the future we may be looking at a different way in which we type blood to get more specific, maybe limit some of the reactions that go on and some of the long-term side effects that can happen. So we have a major histocompatibility complex. These are found on the short arm of chromosome 6. There's about 70 genes, and these code cell protein surface features. They're classified into 3 functional groups. Class 3 genes encode for plasma proteins that carry out nonspecific immune functions. Classes 1 and 2 encode for the human leukocyte antigen, the HLA. So there has been a link with sugars to form the branched glycoproteins that extend from the cell surfaces. HLA glycoproteins recognize bacterial and viral proteins and marking them for the immune response to target. This process is called antigen processing. Class 1 are found on all cells. Class 2 are on our antigen-presenting cells. This is what our antigen-presenting cells look like. We have 2 types. We have the macrophages and the T cells or T lymphocytes. There is some HLA diversity. There are multiple alleles that determine our HLA type. Genetic diversity among HLA genes is very large. 1 in 10,000 unrelated people may share the same HLA type. And matching at least 4 major HLA genes is needed for most transplants. When we think about transplanted organs, bone marrow, those things, for it to be successful. HLA genes count for about 50% of the genetic impact of our immunity. The human immune system, it's a network of vessels and lymphocytes and bean-shaped structures called lymph nodes. The lymph fluid goes through our lymph duct, and it carries our macrophages and our B and T lymphocytes. Organs involved in the production or the maturation of the immune system are our spleen, our thymus, and our bone marrow. And this is just a picture. It's taken from your book to kind of show you how all that looks and where each is kind of released from, and those types of things. So immunity. Immune response attacks the pathogens or cancer cells or even transplanted cells with 2 different types of defense. We have our innate immunity, which is immediate and generalized, and then we have adaptive immunity, which is more specific and slower. They act after various physical barriers block the pathogen. Some of those physical barriers are our first line of defense, such as our unbroken skin, mucous membranes and secretions, the wavy cilia that's in our respiratory tract, the flushing effect of tears or saliva, urination or diarrhea, and we have some non-specific defenses. So the innate immune response is the general defense found in our body. It produces it's a rapid response if a pathogen gets beyond our physical barriers, and the response time is often in minutes. The central part of this is what we see as inflammation, and this creates a hostile environment for the pathogen, and it sends our phagocytes to engulf and destroy the pathogen, and we learned that this was phagocytosis. So levels of immune protection, physical barriers, innate immunity, and adaptive immunity. So the innate immune response collections protects against bacteria, yeast, and other viruses. We have cytokines such as interferon, interleukins, and tumor necrosing factor A. The cytokines play a role in our adaptive immunity, and then we have complement. And this is plasma proteins that assist or complement other defensive responses. The role of complement proteins is that they can puncture bacterial cells, they can dismantle viruses, they can trigger histamine release to dilate blood vessels, and they can also attract phagocytes. Adaptive immunity. This requires stimulation. The response time is generally in days. And the basic characteristics of adaptive immunity include diversity, so there are many different pathogens that can be recognized. There's specificity, so that it distinguishes particular molecules. It has the ability to remember or memory. And then there's the primary immune response, which is what happens at the first exposure. Then we have secondary immune response, and this is what happens at the subsequent exposures, and it uses the memory it's developed from the first exposure. There are two types. There's humoral response, in which our B cells produce antibodies in response to activation by the T cells, and there's cellular, in which the T cells produce cytokines and activate other cells. So here's our humoral immune response. Antigen-presenting macrophages activate the helper T cells. We see that the helper T cells then activate B cells with a matching surface receptors. They divide and produce plasma cells and memory cells. The plasma cells secrete antibodies into the blood that will recognize the antigen presented. And we know that the memory cells remain dormant until subsequent exposures happen, and therefore the response time will be enhanced. The humoral immune response. Different antibody proteins recognize and bind to different features of the foreign cell. Antibody structures minimally consist of 4 polypeptide chains, 2 long, 2 short. There's a constant region, a variable region, an antigen binding site, idiotypes, and epotypes. This is just a picture of antibody surfaces, kind of showing you in pictorial form those definitions that we just kind of talked about on the last slide. What is the function of antibodies? We know that they bind pathogen protein or toxins and inactivates or neutralizes them. They can clump pathogens, making them more visible for the macrophages. They're able to activate complement to boost our innate immune response. An antibody response, however, sometimes can be harmful. Types of antibodies that we have, that would be our IgA, IgD, IgE, IgG, and IgM. And this table kind of tells us what their functions of the different ones are. There are 5 major types. The creation of antibody diversity. The genome has a limited number of antibody genes. So we know that we can shuffle those in many different ways and make seemingly limitless variety of antibodies. Early development of B cells, and there's rearrangement due to enzyme cutting or pasting, which is different combinations of the variable, diversity, and joining. Creates new versions of antibody proteins. This is just kind of a picture of to go along with that. Cellular immune response, maturation of T cells. So we know that the T cells need to recognize the foreign antigen and the not self antigens. We know that the thymocytes can travel to the thymus and display their cellular surface receptors. The thymus lining displays self antigens. T cells bind themselves to antigens and die by apoptosis. And T cells that don't bind and survive, they mature. There are types of T cells. So we differentiate our T cells by the types and patterns of receptors that are on their surface and what their function are. So we have helper T cells. These have our CD4 antigens. We have cytotoxic T cells, CD8 antigens, and our regulatory T cells. What is the role of the helper T cell? It helps in the humoral immune response. It's able to recognize antigens presented by macrophages, and it will also stimulate the B cells to produce antibodies. In the cellular immune response, it can secrete cytokines, and it can also activate our cytotoxic T cell. And this is a type of cytokines, and what are their functions? So we have the colony-stimulating factors, interferons, interleukins, and our tumor necrosing factor. So we have the cytotoxic T cell, which continuously monitors blood cells, recognizing and eliminating our virus-infected and tumor cells. So this is how we see that happen. So we have types of immune system cells. We want to look at macrophages, the dendrite cell, the mast cell, the B cell, the T cell helpers, the cytotoxic T cells. We have neutrophils, natural killers, and suppressors. So when we have abnormal immunity, so as you can see in just kind of overthinking this immune system, it could go wrong in many different ways, and we can have different malfunctions. And those malfunctions can either be inherited, what we acquire from our genetic makeup, or they can be acquired. Your immune system could be too weak, too strong, or be misdirected. The responses could be related to multifactorial issues or caused by a mutation of a single gene. Inherited immune deficiencies are about 20 types. They can affect both our innate or adaptive immunity. Some examples might be chronic granulomatosis disease, which is a mutation of the oxidized enzyme resulting in neutrophils that cannot kill bacteria. Or we can have severe combined immune deficiency, which is the SCID, in which an individual has difficulty in both the humoral and cellular immunity because they lack mature B cells or T cells, or both. So this is the bubble boy. He had SCID, so he had no thalamus gland. And so he had T cells that could not mature and could not activate his B cells. This is what we called the bubble boy. So these are some types of inherited immunodeficiencies. It shows what it is and where the defect is. Quite a few there. So HIV, it's a virus that enters the body with direct contact to bodily fluids. It infects the macrophages and the helper T cells. The virus will replicate and burst out of the helper T cells and kills it. So the loss of our helper T cells prevents our B cells from being activated. The infection occurs because the immune system is rendered not functional, and this virus can replicate rapidly and mutates easily. Here's what happens. So then we have acquired immune deficiency syndrome, which is what we call AIDS, which is the disease that results from the HIV infection. So the impact of the HIV infection progresses to impair our immune function. Genetically diverse populations of HIV in the human host. The treatment requires a combination of medications that have different actions. So drugs can work at different points of the infection. They can work at entry of the virus into the T cell. the drugs can be geared at replication of the viral genetic material, or it can be directed at processing more viral proteins. So here's some drugs that you can get. Proteinase inhibitors, they shorten the viral proteins. Fusion inhibitors, they block the ability to the HIV to bind to the cell. Entry inhibitor keeps the HIV from entering the cell. The CCR5 gene, so this encodes for a receptor protein on the cell membrane, which is a co-receptor for HIV. Individuals homozygous for the 32 base deletion of the CCR5 are resistant to the infection. Heterozygous individuals become infected but stay healthier for several years longer than people who do not have the mutation. So this, you could have this mutation and actually do well if you were exposed to HIV than the average individual without this mutation. Autoimmunity, this is when the immune system attacks the tissue of an individual's own body. The autoantibodies recognize self-proteins. About 5% of the population has an autoimmune disorder. Signs and symptoms will be determined on the cell type, which is under attack. This happens for several ways. Viruses use host proteins on viral cell surfaces. Thymocytes that recognize self-antigens survive instead of self-destructing. Non-self antigens may coincidentally resemble self-antigens. And you can have a skewed X in activation. You can develop this autoimmunity. So some autoimmune disorders. Diabetes is one, hemolytic anemia, multiple sclerosis, rheumatic fever, rheumatoid arthritis, lupus, ulcerative colitis. And you can see where the autoantibodies fight against. So then when we are going to talk a little bit about Rh incompatibility, and this occurs when an Rh negative woman with no Rh factor has a baby that is Rh positive. First, Rh incompatibility, fetal recognizes it as foreign. The mother's immune system attacks the fetal cells and produces a mild reaction. Few antibodies are present. Second, Rh incompatibility pregnancy, there's a larger response, plentiful antibodies, and there can be destruction of the fetal cells and much damage. It's kind of how this works, which is why Rh factor is important for women who are pregnant to know their status. So what is an allergy? An allergy is a immune response to a non-threatening foreign substance called an allergen. They vary in size. Larger ones, grass pollen; smaller ones, cat dunder, dust mites. Asthma is a chronic disease which can result from an allergic response. So allergies require humoral and cellular immunity to be involved. It's your IgE antibodies which are made and bind to the mast cells. And it's these that release the allergy mediators like histamine and heparin that cause the inflammation, runny eyes, nose, rash, and the asthma. The allergen can also activate a specific class of helper T cells, and they release cytokines. Severe allergic reaction throughout the body is called anaphylactic. This shows you when it's first exposed to the allergen, kind of what happens, and then here's what happens after subsequent exposure. Kind of some different things: peanuts, atopic dermatitis, constricted airways, vaccinations. Vaccinations use antigens from a pathogen to invoke immunity within our own body when we're exposed to this. Antigens are chosen to be harmless alone. The ability to respond rapidly to subsequent exposure prevents the infection to agree that it would cause the disease. Vaccine technology goes back to the 11th century, and we know that Edward Jenner used cowpox as the vaccine for smallpox, which was virtually eliminated. This is what smallpox has not naturally infected a human since 1977. So this is one of our, so now we don't vaccinate against smallpox. This is what it used to look like when someone had smallpox. Pretty nasty. So vaccines are mostly injections. However, we're looking at new delivery methods such as nasal spray, genetically modified fruits and vegetables, which you may get your vaccines through. Foreign antigens stimulate phagocytosis to present antigens to nearby T cells. T cells stimulate B cells to make IgA antibodies. And they coat the small intestine and protect against foodborne pathogens. Immunotherapy, medical treatment used to amplify or redirect the immune response. You can use monoclonal antibodies. They're able to detect and target particular antigens, and thus preserving its specificity and amplifying the antibody type. This is what monoclonal antibody technology kind of looks like. Why do we use monoclonal antibodies? We know that it can do basic research or disease diagnosing, home pregnancy tests. Some strips contain anti-HCG monoclonal antibody, which then if you have HCG present in your urine, it'll change the color of the strip. Herceptin blocks the receptors on the breast cells and prevents reception of the cell division signal. So hopefully it decreases the division of the breast cancer cell. Cytokines boost cellular immunity. Cytokines are used to treat a variety of conditions. Some cytokines that you'll see in use are interferon, was used for cancer and multiple sclerosis, interleukin 2, kidney cancer, culinary stimulating factor boosts WECs, especially in individuals with AIDS. Transplantation. This is when organs are moved from one individual to another. This can include the heart, kidney, lungs, and various other organs, and even bone marrow. There are transplants that are performed annually, and recipients are very well benefited, most often from the gain in years of life. Though, however, successful transplants lie in genetics. So there are different types of transplantation that can take place, which is defined by the relationship of the donor to the recipient. So pay attention to these definitions. So this is just a pictorial of those different definitions. Graft rejection. The immune system reacts to the grafted tissue and recognizes it as foreign and will try to destroy it. There is a hyperacute rejection reaction, and this is severe form, the graft rejection reaction, in which the blood supplied to the graft is cut off. There's graft versus host disease, occurs in bone marrow transplants. The immune cells of the grafted bone marrow recognize the host body as foreign, and it attacks the body of the person who received the bone marrow transplant. Genomic view of immunity. We're looking at sequencing genomes of pathogens. This will help us understand how they infect and aid in the development of treatments. Crowd diseases, how do they spread rapidly through unexposed populations? So examples of crowd diseases are smallpox, measles, pertussis, influenza. And bioweapons, bacteria and viruses used throughout history as weapons in their natural state or genetically manipulated. And that's the end.